Okay, so in chapter 13, this is part two, and what I'm talking about in this one now is meiosis and how the alleles segregate and how we actually divide up the genetic material within the cell. So here's a TOK aspect for you guys. So just go ahead and kind of read through this for a second and craft an answer. We'll talk about it a little bit in class, but it says in 1922 the number of chromosomes was counted in a human cell at 48. This remained the established number for 30 years, so that puts it at 1952. And in 1953 is when they discovered, the double helical nature of DNA. So that kind of puts it in a little bit of context for you. For 30 years, people thought that the number was 48, even though if you look back at the photographic evidence from the time frame, it showed that there was clearly 46. So for what reasons do existing beliefs carry a certain inertia? So kind of think about that. We'll discuss it a little bit in class, and then we'll move on. Okay? So as far as the formation of gametes go, when an organism reaches sexual maturity, and in all organisms it's different, the gonads begin producing the haploid cells, either the sperm or the egg, and the process that produces that is referred to as meiosis. And what meiosis allows for is the chromosome number to remain constant from generation to generation. In other words, we have 46 chromosomes. Our grandparents had 46 chromosomes, our great-great-great-grandparents had 46, and so forth. So what the process of meiosis does is it takes the number from 46 in the case of humans to 23, and then when mating occurs, the sperm and the egg unite. 23 and 23 give us 46 again, and then that individual, when it reaches maturity, will begin producing sex cells. Those sex cells will be, having 23 chromosomes in each one of those. So again, just keeps the ploidy the same through the generations. Okay. So the process of meiosis and fertilization is common to all sexually reproducing organisms. And what meiosis again does is it reduces the ploidy, and the ploidy is that chromosome number. The other thing that it does is it provides for some genetic variation, and again, as I mentioned earlier, it keeps the chromosome number the same from generation to generation. So what we're going to do is we're going to break meiosis into two parts, meiosis 1 and meiosis 2. And what you have with meiosis 1 is the homologous chromosomes, those chromosome pairs that we were talking about earlier on, the one from mom and the one from dad. They are actually going to mix in meiosis, but it's going to separate the homologous chromosomes. And then if you recall, during the process of replicating the DNA, when that chromosome gets replicated, it actually has two sister chromatids. So meiosis 1 is going to separate the homologous chromosomes. Sister chromatids will get separated in meiosis 2. So if you look here, meiosis, we said it's going to have the number of chromosomes. And if you look up here at the top, what we've got are the homologous chromosomes right here. And then once the DNA replicates itself, you can see down here we've got the sister chromatids, but these are still homologs, and it's still 2N. Okay, so you've got mom's and dad's copy in the same cell. And then you take this, once you go through meiosis 1, you're going to separate. This is going to move to one end of the cell. This is going to move to the other. So what we've done here now is we've separated the homologous chromosomes. We've reduced the ploidy. We've went from up here a 2N cell to down here 2 1N cells. And then now you can see we've got the sister chromatids. Meiosis 2 is much like a regular mitotic division in that it's going to separate the sister chromatids. So homologous chromosomes separate in meiosis 1. The sister chromatids separate in meiosis 2. So what you're going to end up with, conceivably, are 4 cells from this original cell that we started with. Now in males, you're going to get 4 functional sperm. In female, as you'll see as we talk, this year during this chapter here, and we'll revisit it a little bit next year when we talk about reproduction. With the egg, you actually get 1 egg and then 3 polar bodies. All right. So here's the process of meiosis 1. Just kind of keep in mind some of the things we just learned about with mitosis, in that you get the formation of that spindle apparatus, which you can see kind of looks like a bird cage right here. It's going to grab onto the kinetochore, the centromere, and it's going to pull the chromosomes to the opposite ends of the cell. So as we go through these things, we're going to talk about some of the unique things with prophase 1. This is where you're going to get some gene mixing and so forth. And then here you're separating these out in the anaphase, and then when you get to the next phase of meiosis 2, that's when it's going to look like a mitotic division. So interphase is basically interphase like we've learned. The cell's basically doing its job. In the case of these sex cells during interphase, this is where once you reach maturity, these chromosomes are going to be replicated, the centrosomes are going to be replicated, and the cell is basically going to be preparing itself for division. So with meiosis 1, in prophase 1, what you get are the homologous chromosomes are going to kind of condense. They're going to pair up and form what's known as a tetrad, and this is the phase where crossing over takes place. This is what provides the genetic variation that we see that is unique to the process of meiosis, sexual reproduction. So it's real important that you understand what prophase 1 does, what it accomplishes, and that's gene mixing, and keep that in mind. That's what's giving sexual reproduction the variety that we speak of, which is why we all look different. We're not clones of each other because of what goes on here in prophase 1. Okay. With metaphase now, just like with mitosis, they're going to align at the metaphase plate, and once they align at the metaphase plate, they're going to be pulled apart now. Once the microtubules attach to the kinetochore, they're going to pull the homologous chromosomes apart. So during anaphase now, anaphase 1 is where homologous chromosomes are separated. And they're actually a mix because, again, in prophase 1, you've got a mix, gene mixing occurring, that variation that we spoke of. During anaphase 1 now, this is where homologous chromosomes are separating. So that's another important thing that you're going to want to remember about meiosis. Again, anaphase 1 is where homologous chromosomes are actually separating. Okay? So with telophase now, this is where, the two nuclei are going to potentially reform. You might have some cell resting taking place in this place. Cytokinesis, as you recall from mitosis, is the same thing here. You're going to divide the cytoplasm up, and now what you're going to have are two haploid cells. Okay, they're still going to have the sister chromatids attached, but now the ploidy has been reduced. So meiosis 1 reduces the ploidy. Okay. Now you might have a stage of resting, as I mentioned earlier. We call it interkinesis, and this is where the chromosomes could relax. It could remain in meiosis 1 for a while, etc., or it could go directly to meiosis 2. So with meiosis 2, this is where we're going to separate the sister chromatids. And even though they're haploid cells, it has many characteristics of a typical mitotic division. Okay, and it's going to repeat basically the same things that it just did. If the chromosomes have relaxed, they're going to recondense themselves in prophase. You're going to have the centrosomes moving to opposite ends of the cell. Then we're going to line up at the metaphase plate. We're going to separate the sister chromatids here, and then we're going to give rise to the 4 cells that we've been talking about. Okay, so prophase 2 now, this is where you get the chromosomes condensing again. If they'd relaxed a little bit, the nuclear envelope, if it reformed, it's going to disappear, and this is where the mitotic spindle is going to start to grow and attach to the chromosomes that are there, the genetic material. So now metaphase 2, this is where they're going to align at the metaphase plate, and all the while, this is where the microtubules are going to attach. They're going to kind of have that tug of war that we spoke of when we talked about mitosis. And they're going to the microtubules are going to attach to the kinetochore. And then in anaphase 2, this is where we're going to separate the sister chromatids. This is where we're going to make the chromosome numbers basically the right number in all 4 of the cells that we're producing here. So if you have 4 functional sperm, they're all going to have the correct number of chromosomes. They're going to be haploid cells. They're only going to have 1 of the chromatids in them. If they're an egg, you'll get 3 polar bodies, but in the final functional egg, there's a few things that are going on that we'll talk about later on, but in that final functional egg, it'll be a haploid cell with 1 set of sister chromatids for all of the chromosomes that, when it unites with a sperm, will restore the ploidy. You'll get diploid again, and then the cycle of life will continue. So now telophase, this is where the nuclei are going to kind of reform around them, and this is where you're basically going to kind of put these cells back into a functional order where they're either going to develop into a sperm or to an egg. And then cytokinesis is going to divide the cells, actually, just like, again, rehashing what we learned in mitosis. All right, so in the actual presentation, just kind of watch the movie here, understand what's going on, and just listen to it, think about it. If you have any questions, of course, always write them down. We'll discuss this briefly in class and make sure everybody's questions get answered. All right. So when you compare mitosis and meiosis, the main difference is mitosis is going to occur in the somatic cells, the body cells, and it's going to produce genetically identical cells. And we talked about this with our cancer portion of chapter 12. If it doesn't produce exact clones, then that's what could give rise to a cancer. So with mitosis, we want to produce exact copies of the cells that we started with. But with meiosis, we want to produce genetically different ones. That variety is what's going to be important, and that's what we're trying to accomplish with meiosis, is reducing the ploidy and making genetically unique cells. So if you look here, if you compare mitosis and meiosis, when we look here, we've got all of the homologous chromosomes. You can see here, like this one and this one, these are homologs. This little one and this little one are homologs. This one and this one are homologs. The chromosomes have been duplicated. They're going to all align at the metaphase plate. Okay, these are sister chromatids. And again, when they separate out, you still have 2N and 2N. So in other words, when you look at this, there's mom's and dad's version, okay, mom's and dad's version, and then this one here, mom's and dad's version there. When you look now at meiosis, here in prophase 1 now, this cell moves to prophase 1. This is where these tetrads are going to form. You can see Tetris, like the game Tetris. There's 4 parts in every game piece. Well, there's 1, 2, 3, 4 sister chromatids here. These are going to line up in prophase. In prophase, they're going to line up and cross over. That's where gene mixing occurs. In metaphase, these things align at the metaphase plate. They've already crossed over. They've already gene mixed here in prophase 1. They're going to align. And now in metaphase 1, we're going to separate homologous chromosomes. So if you look here, you can see there's too much genetic information there. All of these have been duplicated. They all look a little different because of the gene mixing that occurred up here in prophase 1. Now we've separated homologous chromosomes. This is a diploid cell with replicated DNA. We're going to separate the homologs. Okay, so this homolog and this homolog are in opposite cells. This homolog and this one are in opposite cells. This one and this one again are in opposite cells. That's why we've reduced the ploidy, okay, in metaphase 1. Now, in the process of anaphase, we're going to move these to opposite ends of the cell. And in doing so, we're going to separate out the sister chromatids, and that's going to be accomplished in anaphase. So homologous chromosomes separate in anaphase 1. We're going to separate these out in anaphase 2. We're going to separate out sister chromatids, and this is what's going to give rise to the 4 functional sperm daughter cells of meiosis, 1 egg and 3 polar bodies, as you'll see, whatever it happens to be. So you can see sister chromatids separate in anaphase 2. Homologous chromosomes are going to separate in anaphase 1. So when we move to from metaphase to anaphase, homologous chromosomes separate. In anaphase 2, sister chromatids separate. Okay, so if you have any questions on those, we can go over that in class as well. All right, so the 3 main events that are important to meiosis, you have synapsis and crossing over. That's the formation of the tetrad that we talked about here, and then the separation of homologs. All right. So in prophase 1, again, these duplicated homologs are going to become physically attached when they line up, forming that tetrad. This is where you're getting the crossing over that's occurring. This is the gene mixing between the non-sister chromatids, and this X-shaped region is what kind of appears. So if you look at it with an electron microscope, you can see this is called a chiasma. This is where you're actually getting physical breakage of the DNA, and they're swapping positions on the actual chromosome, as you can kind of see here in terms of the color. So this is an electron micrograph of actually what's going on. So in prophase 1, now this is where gene mixing is occurring, and then they're going to line up in metaphase. They're going to separate out. Homologous chromosomes will separate in anaphase 1. Sister chromatids will separate in anaphase 2, and then this is what's going to give us 4 daughter cells of meiosis. It doesn't matter if they're sperm or egg really at this point, but you can see how they're all different. Okay, they're all potential mixtures of the original kind of copies that we started with up here. Okay, so now the tetrad formation, this is what we were talking about. This is where you're getting 4 things kind of aligning. And if I back up, this is the tetrad here. You can see it's kind of underlined there. That's the actual tetrad that we're talking about. That's unique and important to meiosis. And then those tetrads are going to align after crossing over has occurred. And then that's where the homologous chromosomes are going to be separated in anaphase 1. So this is the other important thing here with the separation of the homologs. In anaphase 1 of meiosis, this is where the duplicated chromosomes are going to move to the opposite poles. So the sister chromatids at this point, because we're in metaphase 1, meiosis 1, at this point the sister chromatids are still attached to one another. Okay. So 3 of the mechanisms now that produce genetic variation that is unique to sexually reproducing organisms, and this is the important part about sexual reproduction, is you have independent assortment, you have crossing over, and you have random fertilization. So these 3 things are what produce the genetic kind of variety that we see. So homologous chromosomes can be positioned independently. It's just a flip of a coin. They can be this way or they can be this way at the metaphase plate. There's no rhyme nor reason to how they line up. And the maternal and paternal chromosomes, they assort independently of every other pair. There's no rhyme nor reason. This is just like a roll of the dice, so to speak. And these things line up how they line up. So we have 23 pairs of chromosomes that are all lining up any random number of ways. There's over 8 million different ways that these chromosomes can line up. So it's kind of crazy when you think about this. Just independent assortment alone can give rise to 2 to the 23rd, because there's 23 pairs of chromosomes, over 8 million different combinations in humans. Okay, so that right there is a big number. And then now crossing over, this is gene mixing. So now with the gene mixing between the non-sister chromatids, this is where you're getting the swapping and the mixing of the genes, the creation of this variation that we've been talking about. So now when you're crossing over about 20,000 different genes, that's really, adding to the variety that we're talking about, in addition to the 8 million ways that we said that they can line up. So you can start to see now how this number is getting almost ridiculous in terms of the number of different combinations that you can have. And then when you talk about the idea of random fertilization, when you have the one egg and then you have hundreds of millions of sperm that could potentially fertilize that egg, the odds of having one egg be fertilized by a sperm and then a second egg where all of the processes have been exactly the same. Okay, of course that's not going to happen, but having another egg that's exactly like one that was created prior to that, and then of those hundreds of millions of sperm that are being released during mating, you've got the same thing happening in that sperm. Now that sperm has to find an egg where it occurred. So you can just see how ridiculous it is. And that's why nobody really looks the same unless they're identical twins. They might have a lot of striking features and similarities about them, but because of just the math of all of this involved here, with all of the endless combinations and ways that these things can line up, and then the sheer numbers that we're talking about, nobody is ever going to look exactly like somebody else. So if you want to look at how variation is kind of created, you can watch this video here in the presentation. Just follow the link. And then again, as is with the case with all of these things, as you have questions, please make sure that you write them down, star them in your notes, and then ask. Okay? So I hope this helped, and good luck. See you in class.